So let's start. Firstly, we are doing 1 mark questions. Okay. The very first question is protein P becomes functional upon phosphorylation of serine. Replacing this serine with dash dash will result in the phosphomimic mutant of P. Our answer should be phosphomimic mutant of P means same or the copy of phosphoserine, which is asparic acid. The answer is option number B, asparic acid. The next question is RAS protein is a, which is state theory question. The answer should be monomeric GTPase involved in relaying signal from the cell surface to nucleus. Option number B. Okay. Let's move to the next question. Which is of the following statement are correct? Viruses can play a role in causing human cancer, which is true. A tumor suppressor gene can be turned off without any change in its DNA sequence. It's also true. Alternation in miRNA expression level contributes to the development of the cancer. This is also true. Then the answer should be PQR. Option number D. The next question is, which class of antibody is first made by developing B cells inside the bone marrow? IgG, IgE, IgA or IgM? The answer should be IgM because IgM always gives primary response and it's primarily produce antibody. Okay. The next question number 5. Determine the correctness or otherwise of the following assertion and the reason regarding mammalian cells. Okay. So they have given a sentence and the reason. We have to find the sentence is correct or not and the reason given is right according to the sentence. Okay. Let's read the sentence firstly. Cells use Ca2+ and not Na+ for cell-to-cell signaling, which is completely true. Okay, and the reason is in the systole concentration of Na+ is lower than that of Ca2+. Okay, which is not true because it's always higher than that of Ca2+. Okay, then the option should be option number D. A is true, but R is false. Okay. Let's go to the next question. Question number 6. Vincristine and vinblastine, two commercially important secondary metabolites from Catharanthus roseus, are the example of not steroid, not terpenoids, not flavonoids, always alkaloids. Okay. Vincristine and vinblastine are the examples of alkaloids. Let's move to the next question. DNA synthesized from an RNA template is called as, it is straight question or the theory question. It's called as complementary DNA. Okay, everyone should know that. Question number 8. Two monomeric his stack proteins. Okay, so now let's pause the video and read the question. I hope you did it. Okay. There are two proteins having same molecular weight in a solution. We have to separate them from the solution. Which of the following technique we can use? Let me tell you. Ion exchange chromatography is the technique we can use to separate two proteins. Okay. Question number 9. During positive negative selection process, transform animal cells expressing dash dash are killed in presence of ganciclovir in the medium. The answer should be viral thymidine kinase. Option number B. Let's move to the next question. A vector derived from which of the following viruses is used for high-frequency genomic integration of transgene in animal cells? The answer is lentivirus. Okay. Which one of the following statement about Agrobacterium TI plasmid is correct? Let's go one by one. Which genes are located within the tDNA segment, which is false, is located outside the tDNA segment. Phytothrombin biosynthesis genes are located outside the tDNA segment, which is also false statement. Opine catabolism genes are located within the tDNA segment, which is also wrong. It's located outside the tDNA segment. So we have only one option remain. Opine biosynthesis genes are located within the tDNA segment, which is true statement. Okay. Question number 12. Which of the following types of molecules are act as a biological catalyst? Or we can say that they are act as an enzyme. Okay. So proteins are act as a biological catalyst. We know that in RNA. RNAs also act as a biological catalyst. Ribozymes, ribonucleic acid enzymes, okay, are RNA molecules that have the ability to catalyze specific biochemical reactions, including RNA splicing in gene expression. Okay, phospholipid, which is not act as a biological catalyst. So the answer should be option number A, P and Q only. Let's move to the next question. Which of the following media components used in maintain pH in mammalian cell culture? Okay, the answer should be NaSCO3 because it acts as a buffer, therefore it used to maintain pH in mammalian cell culture. Question number 14. Which of the following are energy transducing membranes? Plasma membrane of bacteria. Yes. Inner membrane of fluoroplasm. No, it's not energy transducing membrane. Next is inner membrane of mitochondria. Yes. So the answer should be option number B, P and R. The next question, let's move to the next question. Pause the video and read the question carefully. Okay. Same type of question we have touched earlier, which is assertion and reason. Let's see. The assertion which is given means sentence is the two trees will not be identical. Yes, which is true. The nature of frequency of mutations in two families are different. So this is also correct. Okay. So the option should be both A and R are true, and R is correct or correct reason for A. Okay. Let's move to the next question. Question number 16. A microorganism isolated from salt-rich lake was found to possess diglycerol tetraethers with polyisoprenoid alcohol side chain as the major lipid component of its cell membrane. The isolated organism is, the option answer is option number D, Archaea. The next question is, the composition has given for carbon it's 1, hydrogen is 1.6, for oxygen is 0.4, for nitrogen is 0.2, for sulfur is 0.0024, and for phosphorus is 0.017. We have to find the biomass of carbon. Okay, atomic weights are also given. Okay, now just multiply each composition with their atomic weights, then add them. Okay, like this. Multiply their atomic weights, okay, with composition and then addition of that multiplication. And for the contribution of carbon in percentage, we just have to atomic weight divide by the total we have got into 100. Okay, so the atomic weight of carbon is 12 divided by the total and into 100. Then we'll get our answer, which is 51.2. Okay, got it? So the next question is, pause the video and read the question carefully again and again. Okay, I'll give you the formula how to solve the question. Okay, we just have to get total and the solubility of A. Okay, and then we'll get the answer, which is 60 gram. The next question, the number of molecules of nucleotide of molecular weight 300 gram per mole present in 100 picomoles is dash dash. Okay, directly calculate the number of molecule. The 10 picomole, we know that. Okay, the 10 picomole is equal to 10 into 10 raised to minus 12 mole, and 1 mole is equal to 6.023 into 10 raised to 23. Nothing had to do, just multiply both. Okay, which will come 10 into 10 raised to 12 into 6 into 10 raised to 23. We'll get our answer, which is 6. So the next question, read the question carefully, pause the video. Let me give you the formula for that, answer will come. Okay, write the formula carefully. We have to find the value of n. So the formula should be n N is equal to A divided by pi D to the base B square. Okay, we'll get our answer which is 2. Okay, from now each question carries 2 marks. Okay, a block of ice, let's start the question number 21. A block of ice at 0 degrees Celsius supplied the heat at a constant rate. To convert ice to superheated steam, which one of the following trajectories correctly represent the trend of the temperature of system with time? Assume that specific heat of H2O is not the function of temperature. The correct answer is option number A. Okay. The next question. The DNA sequence shown below in the amplified PCR. Okay. So they have given a particular sequence. We have to find which pair of the primer we can use for amplification. For amplification, we have to use that primer, which will be option number B. Okay. Question number 23. Which of the following statements about immune response are correct? Okay. The T cells activated by, first is the T cells activated by antigen presenting sig, which is true statement. Okay. The foreign peptides are not pretended to help a T cell by class 2 MHC proteins. Okay. This statement is incorrect. The third one is dendritic cells are referred as a professional antigen presenting cell. This is 100% true. Okay. Then our answer should be option number 1, P and R only. So the next question, which of the following statements are correct about eukaryotic cell cycle? Let's go one by one. The first is CDKs can phosphorylate proteins in the absence of cyclins, which is false. Okay. Next is CDKs can be inactivated by phosphorylation. Yes, it's true. Dehydration of cyclin is required for cell cyclation progression. Okay, which is correct. CDKs are not involved in chromosome condensation, which is not correct statement. Then our answer should be option number D, Q and R only. Next question. Read the question, pause the video, read again and again, understand what is given. So they have given a biochemical pathway which is SWXYZ, which is in the form of SWXYZ. Read again and again. The answer is question option number B. Okay, option number B is correct answer. The next question. Pause the video and read the whole question. There is a tomato plant, red and yellow, it's food color, and purple and green, it's stem color. We have number of progenies plant. Okay, we have to find the genotype of the parent plant. Okay, red is dominant over yellow and purple is dominant over green. Okay, so let's first do for red fruit and yellow fruit. The addition should be 329 and its addition should be 111. Okay, we say that average ratio is 3:1. Okay, 329 and 111, we can say that average ratio is 3:1. Then do same thing with for purple stem and green stem. For purple stem and the green stem, add them both, we'll get the ratio 1:1. Then answer should be option number A. Okay, the next question is 27. Some of the cytokinins used in plant tissue culture media are given below. Which of these are synthetic analogs? We have to say that. The first one is BAP, okay, which is first generation synthetic cytokinin that inhibits plant growth and development. Okay, the second is zeaxanthin, it is not. The third is kinetin. Yes, it is a synthetic compound that regulates cell growth in plants. Okay, and the third is 2-IP. Okay, which is not a synthetic analog. The option should be P and R. Question number 28. Carl Wood used the gene sequence of which one of the following for phylogenetic taxonomy of prokaryotes? The answer is B. A ribosomal DNA of small ribosomal subunit. So question number 29. We have to match the pathogens given in group A with their antimicrobial agents in group 2. Okay, the first is influenza A virus. Its agent is amnetadine. Okay, the second is fungus whose agent is fluconazole. Okay, the third is plasmodium. Its agent is artemisinin. And the fourth is mycobacterium. Its agent is isoniazid. Okay, so the answer should be option number C, P2, Q3, R4 and S1. Question number 31. Okay, again assertion reason question. Assertion is DAM methylase protects E. coli DNA from phage endonucleases. Okay, and the reason is E. coli DAM methylates the adenosine residue in the sequence GATC. The answer is option number D. A is false but R is true. Question number 31. Again assertion and reason. Assertion is embryonic stem cells are suitable for developing knockout mouse, and the reason is homologous recombination is more frequent in embryonic stem cells. The answer is option number B. Both A and R are true, and R is correct reason for A. A schematic of plasmid with a gap in one of the strands is shown below. Which of the following enzyme is required to fill the gap and generate a covalently closed circular plasmid? Okay, the first is DNA ligase, which is required. Okay, second is alkaline phosphatase. It's not required because OH group is present at 5'. Third is DNA polymerase, which is required, and the fourth is polynucleotide kinase, which is also required. So the answer should be option number B, P, R and S only. Question number 33. Group A has cellular organelles and we have to match them with their features in group 2. The answer is C. Okay. The mitochondria has maternal inheritance. Chloroplast has endosymbiotic origin. Nucleus has double membrane enclosed, and the endoplasmic reticulum is single membrane enclosed. Okay. The next question is question number 34. Which of the following strategies are used by cell metabolic regulation? First is phosphorylation, dephosphorylation. Second is allostery, and the third is feedback inhibition. The answer is option number D. All the strategies are required. All the strategies are used. You can say. Question number 35. Determine the correctness or otherwise assertion and reason question. The assertion is a zygote and its immediate descendant cell are unspecialized and are called totipotent. And the reason is totipotent cell retain the capacity to differentiate into only a few cell type. The answer is option number D. A is true but R is false. Question number 36. Which of the following statements about gene therapy are correct? Okay, pause the video, read all the statements one by one. The answer is correct statements are Q and R. are both correct statements. Q and R. The next question, the last question. Determine the correctness, assertion and reason. Okay. Assertion is a genetically engineered rice that produces beta carotene in the rice grain is called golden rice. And the reason is enabling biosynthesis of provitamin A in the rice endosperm, which a characteristic yellow or orange color. The answer is option number C. Both A and R are true and R is correct reason for A.